minds? Get ready for an adventure as we navigate the complex highways of how our cells make and regulate energy. Today we are diving into carbohydrate metabolism, a crucial process that keeps living beings lively and kicking. It's like a lively dance of biochemical reactions, making sure our cells have the energy they need for all their daily tasks. Let's break it down and uncover the secrets that keep life ticking along smoothly. Let's explore 4 important aspects of carbohydrate metabolism together. We will start by understanding how our cells make energy from glucose. Then move on to the clever ways our bodies store and use glycogen. Next, we will look at how our bodies can create new glucose when needed even without eating it.
Finally, we will pick up some key terms to understand the language of glucose pathways.
It's like unraveling the secrets of how our bodies turn food into energy.
Okay.
In utilizing the sequence of Krebs cycle to oxidize glucose, it consists mainly of a 10-step enzymatic process of glycolysis that converts a 6-carbon glucose molecule into 2 or 3 molecules of pyruvate. During this process, we will produce 2 ATP and 2 NADH. The pyruvate molecules must be moved from the cytosol into the mitochondrial matrix, where our 3-carbon molecule will further oxidize to become acetyl-CoA, which is a 2-carbon molecule. As a result, it will release 2 carbon dioxide molecules into the environment for every 3-carbon glucose molecule.
Nice.
Let's explore how glycogen functions as an energy reserve in our bodies. Glycogen serves as a storage form for glucose, primarily located in the liver and muscles. While liver and glycogen caters to the needs of both itself and the body, muscle glycogen is exclusively available for the muscle use. It's important to recall that glycogen stores have a limit. After a meal, glucose initially replenishes these stores and then transforms the remaining excess, When physical activity begins, there is a quick demand for ATP, met by phosphocreatine and glycolysis. Consequently, glycogen becomes a local, immediate, and rapid source of glucose. Glycogen is a glucose polymer with a highly branched structure, featuring two types of linkage, the alpha 1 to 4 linkage forming a linear chain and the alpha 1 to 6 linkage creating bunches. Stored in the cytoplasm, glycogen is typically associated with multiple proteins. Glycogenolysis or glycogen degradation involves the removal of glucose molecules from non-reducing ends. Given its multiple non-reducing ends, glycogen can supply glucose rapidly. In glycogenesis, the synthesis of glycogen, glucose transforms into glycogen using UDP glucose as the building block and UDP as the energy. Glucose undergoes several steps, with glycogen synthase being a key enzyme. However, the provided diagram is a simplified summary that omits some details. Hexokinase initiates glycolysis by converting glucose to the glucose 6-phosphate. Subsequently, phosphogluconeogenesis transforms glucose 6-phosphate into glucose 1-phosphate, leading to glycogen synthesis, glycogen degradation, or glycogenolysis. It involves multiple steps to produce glucose 1-phosphate with glycogen phosphorylase as the primary enzyme. The process then reverses, converting glucose 1-phosphate back to glucose 6-phosphate via phosphogluconeogenesis. In muscles, glucose 6-phosphates proceeds to glycolysis, while in liver, it can be converted back to glucose by glucose 6-phosphatase for release into the bloodstream. That is all. Thank you.
Oh my god! Wow!
Now, let's talk about gluconeogenesis, a carbohydrate metabolism. Under normal conditions, carbohydrates are broken down by various enzymes into glucose, but under special circumstances, substances like fat, amino acids, and pyruvate are converted into glucose. This process is essential for maintaining blood glucose levels between 100 to 200 milligrams per deciliter throughout the day. During fasting, excess glycogen is stored in the liver, which is used later when blood glucose levels fall. However, certain tissues in the human body, such as the nervous tissue and red blood cells, can only utilize glucose as a source of energy. Substances like fat, amino acids, and pyruvate are converted into glucose by gluconeogenesis, which maintains blood glucose levels. The metabolic reactions involved in gluconeogenesis include glycolysis, the Krebs cycle, and sub-monthly reactions. The process involves a series of 10 reactions, with 3 irreversible reactions being catalyzed by specific enzymes. These reactions are upregulated only when glucose concentration is high, preventing excess glucose from being converted to pyruvate. However, these reactions can be reversed using specific enzymes, which are upregulated only when there is a deficiency of glucose in the blood. Glucose is converted to glucose 6-phosphate by the enzyme glucokinase, fructose 6,6-phosphate by the enzyme phosphofructokinase, and fructose 16+-phosphate by the enzyme phosphoenopyruvate. The less irreversible reaction is the conversion of glucose 6-phosphate back to glucose by the enzyme glucose 6-phosphate. Pienate is first converted to succinyl-CoA, which is then converted to oxaloacetate and phosphoenopyruvate. Lactate is converted directly into pyruvate by the enzyme lactate dehydrogenase,
and glycerol is converted to glycerol 3-phosphate, which is then converted to dihydroacetone phosphate. In conclusion, gluconeogenesis is a crucial process that helps maintain blood glucose levels by converting carbohydrates into glucose.
Okay.
I will discuss about terminology of glucose metabolic pathway. I will teach the connections between four frequently occurring metabolic pathways that involve glucose. On the left side, it is important to note that the pathways responsible for glycogen degradation are named with suffixes such as lysis, indicating breaking down or splitting. Conversely, on the right side, the pathways associated with glycogen synthesis are named with suffixes like genesis, signifying the process of making or formation.
Let us now present the definition of each two, starting with glycolysis, also known as the EMP pathway or the Anden-Mayer-Hofmans pathway, was discovered by 3 individuals.
It is a metabolic pathway that converts glucose into 2 molecules of pyruvate, producing ATP as a form of chemical energy and generating NADH-reduced coenzymes. Glycolysis occurs in the cytosol or cytoplasm and is common to both aerobic and anaerobic respiration. Moving on to the next term, gluconeogenesis is the metabolic pathway responsible for synthesizing glycogen from glucose 6-phosphate. This pathway involves the conversion of excess glucose into glycogen by liver cells, aided by the insulin hormone. Glycogenolysis, on the other hand, is the metabolic pathway that breaks down glycogen into glucose 6-phosphate. Liver cells play a crucial role in this process as they convert glycogen into glucose with the assistance of the glucagon hormone secreted by the pancreas. Lastly, gluconeogenesis is the metabolic pathway that synthesizes glucose from non-carbohydrate materials such as amino acids, fatty acids, and glycerol. This process primarily takes place in the liver and kidneys. I created a table here that provides a concise explanation of the terms. This will help you easily differentiate them and become familiar with their processes. Let's start with glycosis, where glyco refers to glucose, meaning sweet. Its function is to convert glucose into pyruvate. Next, we have glycogenesis, which involves the synthesis of glycogen from glucose. Genesis means creation in Greek. Moving on to glycogenolysis, glycogen refers to the composition, and its function is the breakdown of glycogen into glucose. Lastly, we have gluconeogenesis, where glucose and neo mean glucose. Its function is the synthesis of glucose from amino acid, pyruvate, and other non-carbohydrates. These are the shortened definition of the other 4 terms in the table.
Okay.
In summary, carbohydrate metabolism is integral to cellular energy provision and glucose regulation, optimizing ATP production through glucose oxidation. The dynamic interplay of glycogen synthesis and breakdown offers a responsive energy storage system. Additionally, gluconeogenesis allows the synthesis of glucose from non-carbohydrate sources, crucial for sustaining energy during fasting or low-carb states. Mastering the associated terminology is the key to understanding the intricate molecular events orchestrating energy metabolism. Overall, these processes showcase the remarkable adaptability and precision of cellular mechanism in meeting the diverse energy needs of living organisms.